Welcome friends to a new session on genetics. In this session, we will discuss extensions to Mendelian genetics, gene interactions and modified dihybrid ratios. Extensions to Mendelian genetics, gene interactions and modified dihybrid ratios. Let us first see about interaction of genes. At the beginning of the 20th century, geneticists had no idea about how genes influence particular phenotypes. They knew nothing about the structure and function of genes or any techniques to study about it. They proposed everything about the action of genes by analysis of phenotypes. Mendel's experiments suggested that each character is controlled by a single factor or a single gene. The gene is a chemical determiner which is located on chromosomes. The chromosomes comes in pairs. So, the genes also inherited in pairs and they assort independently. Law of independent assortment states that when the parents differ in two or more alleles, then the inheritance of allele is independent of others. The offspring receives one gene in each pair of genes from each parent and they segregate independently. Law of segregation states that when a pair of contrasting alleles comes together in an organism, they remain pure and separate again in the gametes. One of two or more versions of a gene is called allele. An organism carries two copies of the gene, or if the genes are the same, it is referred to as homozygous, and if the genes are different, it is referred to as heterozygous. Typically, the recessive gene is suppressed by the dominant gene in the heterozygous condition. The gene expresses either in the heterozygous state or in the homozygous state is known to as dominant factor. A gene that can express itself only in the homozygous state is referred to as a recessive factor. A key concept in classical genetics and Mendelian inheritance is dominance. According to Mendel, the law of dominance or law of purity of gametes stated that the recessive alleles will always be masked by dominant alleles. To study all this, he developed test cross and dihybrid crosses. The significance of a test cross is to determine if this individual is homozygous dominant or heterozygous. A dihybrid cross is a cross between two individuals that are both heterozygous for two different genes. Mendel used dihybrid to formulate his law of independent assortment. Anyway, the concepts behind a test cross are important. One of the important findings of Mendel's experiments is that genes can exist in alternate forms. He identified two different allelic states, one dominant and one recessive. When dealing with a trait for which there is a dominant gene and a recessive gene, three combinations or three conditions of the genes in the gene pair can occur. They are: 1. The homozygous dominant conditions occurs when both genes present in the dominant state. 2. The homozygous recessive condition occurs when both genes present in the recessive state. 3. The heterozygous condition occurs when one gene present in the dominant state and the other in the recessive state. But not every trait is controlled by a single pair of genes. But later discoveries proved that the interaction of more than one pair of genes controls a single phenotypic character. This is known as interaction of genes or factor hypothesis. The interaction of genes or factor hypothesis was proposed by William Bateson. He is an English genetist and a fellow of St. John's College, Cambridge. He was the first person to use the term genetics to describe the study of heredity and biological inheritance, as well as the chief popularizer of the ideas of Gregor Mendel following their rediscovery in 1900 by Hugo de Vries and Carl Correns. He, along with Reginald Crundell Punnett, a British genetist who co-founded with William Bateson the Journal of Genetics in 1910, and he is also the creator of the Punnett square. A gene interaction is the interaction of genes on a specific phenotypic character. The gene interactions occurs in between the genes located on the same chromosome or different chromosome is known as intergenic or non-allelic gene interaction. The gene interaction occurs in between the two alleles of a gene called intragenic or allelic gene interaction. A new phenotype was produced in intergenic interaction. There are various types of interactions, and these interactions leads to a different range of phenotypes. Examples for the most important types of gene interaction are comb pattern in poultry and flower color in sweet pea, which is called as Lathyrus odoratus. Let us first examine about the inheritance of comb pattern in poultry. Some of the first evidence that a trait can be influenced by more than one gene was obtained by Bateson and Punnett from breeding experiments with chickens. They clearly demonstrated that two independently assorting genes can control a single trait. That means different combinations of alleles from the two genes result in a different phenotype. Bateson and Punnett analyzed the three comb pattern of chicken known to exist at that time. Chickens have a variety of comb types. They are rose, walnut, pea and single. Rose comb found in Wyandotte breed, pea comb in Brahma breed, single comb in Leghorn breed and walnut comb found in Malay breed of fowls. Rose comb is a classical monogenic trait of chickens and varies in different breeds. The rose comb is a fleshy, tube-shaped solid comb that extends from the beak to the back of the head with a pointed spike. Top surface is slightly convex and with small rounded protuberances. The front portion of the comb is covered with small round bumps terminating in a tapering spike which may turn upward, follow the contour of the head in Wyandotte's or nearly horizontal as in rose comb leghorns. The pea comb is a medium sized, smooth and soft comb having three ridges. The middle ridge is a bit higher, running from the beak to the top of the head, which are either undulated or marked with small rounded serrations. The single comb is the most widely recognized type of chicken comb and is rigidly upright. It is attached to the chicken's skull from the beginning of the beak to the back of the head and features a series of 5-6 fleshy ridges called points and a thicker portion called the blade in the back, often more typical of hens. The walnut comb gets its name from the characteristic furrowing in its surface and is similar to the outer appearance of a walnut shell. It is a medium sized solid comb genetically derived from two dominant alleles for the P and Ross combs typical of silky chickens. The development of comb is controlled by two independently assorting non-allelic gene pairs capital R and capital P on two different chromosomes. The gene capital R gives rise to Ross comb and capital P produces P comb. A cross was performed between Ross represented by capital R, small p pattern bearing fowls and single represented by small r, capital P pattern bearing fowls. The capital R and capital B gene for Ross and P combs together produce a new phenotype walnut represented by capital R, capital P in the F1. In the heterozygous state, both the P comb gene and the Ross comb gene can express themselves. That is, only one copy of the Ross comb gene or the P comb gene is sufficient for that type of comb to occur. Therefore, both genes in this case act as dominant genes. When the F1 hybrids are interbred, an interesting result was obtained. The F2 showed walnut, Ross, P and single comb in the ratio of 9:3:3:1. In this, the F2 ratio is not altered, but it is the clear indication of involvement of more than one pair of genes. In F2, the P comb gene is absent and one copy of the Ross comb gene is present. It results in Ross comb. In other words, a Ross comb has two possible gene combinations: capital R, small p, or capital R, small p. When the Ross comb gene is absent and one copy of the P comb gene is present, the result is a P comb and has one of two possible gene combinations: small r, capital P, or small r, capital P. One copy of each gene is present in chicken. It results a walnut comb and has one of four possible gene combinations: capital R, capital P, capital R, capital P. Third possibility is capital R, capital P, or capital R, capital P. When both genes, that is capital R and capital P genes, are absent, it results in a single comb and has only possible gene combination small r, small p. In this experiment, the single comb acts as recessive to both capital R gene and capital P gene, indicating that the single comb have recessive alleles for Ross and P. The recessive alleles for both Ross, P in single comb indicates that more than one pair of alleles controlling comb shape. In F1, the production of walnut comb clearly indicates the interaction of two dominant genes, capital R and capital B, for Ross and P. In F2, Ross comb pattern is produced by capital R gene together with small p gene, and the P comb is controlled by capital P gene with a recessive small r gene. Single comb is produced by the genes capital R and capital P in their recessive state. In this experiment, the inheritance pattern of genes does not differ from Mendelian pattern. The main difference is new character appear in the form of single. Now, let us see about complementary genes. Complementary genes may be defined as two or more genes occurring in different loci of the same chromosome, or different chromosomes interact with each other to produce a character, but neither of them produces that character in the absence of the other, and the action of these independent genes are complementary. In this type of gene interaction, the production of a new phenotype requires the presence of dominant alleles of the both genes from the parents, which controls the character. When any one of the two or both genes are present in the homozygous recessive state, the contrasting phenotype is produced. Thus, any one of the two dominant genes is unable to produce the phenotype when they are alone, but they complement each other to produce the concerned phenotype when they are together. For example, flower color in Lathyrus odoratus, which is the sweet pea, is determined by two genes. In this case, the genotypes capital C, small a, otherwise capital C, small a, yet another genotype small c, capital A, another one small c, capital A, and small a, small a produce one type of phenotype because of the complementary action of these genes in the F2 ratio 9:3:3:1 would now become 9:7. Now, let us examine in detail about flower color in Lathyrus odoratus. Betson and Punnett in 1904 studied the inheritance of flower color in sweet pea Lathyrus odoratus. Sweet pea is an annual climbing flowering plant to a height of 1 to 2 meters, where suitable support is available. It belongs to the genus Lathyrus and the family Fabaceae. The plant is native to southern Italy, Sicily and the Aegean islands. They opted to use sweet pea plants because it is relatively easy to perform hybrid crosses with these plants, and chose to focus on the characteristics of flower color. There are two varieties of pea plant, one producing red color and the other producing white color. They selected two white flowered true breeding varieties of sweet pea plants with genotypes capital C, small a, and small c, capital A, respectively, for the study of inheritance pattern in sweet pea. When two true breeding pure line varieties of white colored sweet pea plants with genotypes capital C, small a, and small c, capital A were crossed, the F1 progenies produced red flowered plant with the genotype capital C, capital A, small a. In F2, the F1 plants were self-pollinated among themselves. In F2 generation, the offsprings resulted in the ratio 9:7, of which 9 were red, 7 were white. In F2 generation, out of the 16 progenies, 9 showed phenotypically red flower and 7 appeared as white flower. From the above experiment, it is to be noted that red flowered sweet pea plants with both complementary capital C and capital A genes were in 9 progenies, whereas white flowered sweet pea plants in which either of the complementary gene capital C or capital A is present are 7 in number. After many years, Bateson first described this modified 9:7 phenotypic ratio in sweet pea plants. Researchers were finally able to determine the two genes capital C and capital A responsible for it. The surprising results can be analyzed by understanding the mechanism of color production in flowers. The table shows the 9:7 ratio is a modification of not genotypic but phenotypic Mendelian ratios. Note that the capital C and capital A genes are sought independently, and note that the presence of a recessive genotype at one locus, that is small c or small a masks the effects of the alleles at the other locus. It is obvious that both the dominant alleles capital C and capital A is necessary for the production of pigment in flowers. There are 7 combinations of either a small c or small a which would yield the white flower phenotype. Hence, the 9:7 phenotypic ratio of purple to white flowers. The cross can be represented as follows. The above experiment showed that the red colored flower is due to the interaction of two complementary genes capital C and capital A, but the white colored flower is lacking either capital C or capital A genes in the genotype of the offsprings. In the F2 generation, the pea plants with both capital C and capital A genes are present together in the genotype, which produces the red coloration due to the phenotypic expression of dominant gene capital A along with the complementary capital C gene. The interesting progenies in the F1 and F2 generation were analyzed for the mechanism of color production in pea plants. These genes capital A and capital C control sweet pea flower color by controlling pea plant biochemistry, in particular that related to pigment compounds called anthocyanins. The genetic analysis showed that the color in flowers is due to the action of the pigment anthocyanin. In peas, there is a two-step chemical reaction for the formation of anthocyanins. Gene C is responsible for the first step and gene A is responsible for the second. If any step is non-functional, there is no purple pigment is produced and the affected sweet pea plant bears only white flowers. The dominant capital C and capital A alleles code for functional steps in anthocyanin production, whereas the recessive small c and small a alleles code for non-functional steps. Thus, white flowers will result if two recessive alleles occur for either gene. Here, thus capital C gene is responsible for the production of the enzyme that catalyzes the necessary chemicals for production of an intermediate complex in anthocyanin pigment formation, whereas gene capital A is responsible for production of enzyme which catalyzes the intermediate colorless complex into anthocyanin, the color producing pigment. Therefore, both capital C gene and capital A gene have their combined effect in production of anthocyanin pigment, but alone capital C gene or capital A gene cannot help in production of anthocyanin pigment. Let us summarize the foregoing discussion of gene interactions. Genetic analysis can determine that the gene interactions determine the phenotype of an organism. According to Mendel, each character is controlled by a pair of genes, but later discoveries proved that a single character is controlled by more than one pair of genes, known as interaction of genes or factor hypothesis. Gene interaction is of two types, allelic gene interaction and non-allelic gene interaction. Allelic gene interaction occurs between the two alleles of the same gene, but non-allelic gene interaction occurs between the alleles of different genes. Inheritance of COMs and complementary genes are the most important type of gene interaction. Flower color in sweet pea is an example for complementary genes. The work of Bateson and Punnett in comb pattern study in chicken and flower color in sweet pea clearly demonstrated that two independently assorting genes can control a single trait. In this experiment, the inheritance pattern of genes does not differ from Mendelian pattern. The main difference is new character appear in the form of single. In flower color in sweet pea, the 9:3:3:1 ratio is modified into 9:7. The surprising results can be analyzed by understanding the mechanism of color production in flowers. The color in flowers is due to the action of a pigment known as anthocyanin, and here the gene capital C is responsible for production of enzyme responsible for pigment production. Phenotype expression in color variation is due to presence of dominant alleles of both the genes or complementation of gene action. Now let me give you some assignments based on the discussion what we had. The first one is explain how genes and their interactions produce a range of different phenotypes. Second is who proposed interaction of genes or factor hypothesis. Explain different types of gene interactions. Third assignment would be explain the gene interaction in flower color in sweet pea. Fourth, what is the key concept in Mendelian inheritance and classical genetics? Explain. Fifth, explain dominant and recessive factor. Now let me give you some references for further reading. The book with the title iGenetics, a molecular approach plus mastering genetics with e-text, 3rd edition, authored by Peter J. Russell, published by Pearson in 2011. Second, a book with the title An Introduction to Genetic Analysis, 7th edition, authored by Griffiths, Miller, Suzuki, Levontin and Gilbert. It is published by Freeman, New York in 2000. A third book is with the title Principles of Genetics, 8th edition, authored by Gardner, Simmons and Nestad in the year 2006 and published by John Wiley, New York. A fourth book is with the title Concepts of Genetics, 10th edition, authored by Klug and Cummings in the year 2011 and published by Benjamin and Cummings. A fifth book is with the title Principles of Genetics, authored by Gardner and Nestad in the year 1984, published by John Wiley, New York. Let me also give you some websites for your further reference. One is www.genetics.org. Yet another would be theagricos.com. Thank you for watching this session. See you next time. Until then, bye.